Hello, everybody. Welcome to NannoTalk. Today, we are pleased to have Rosie Sheward working at the Goethe University in Frankfurt, in Germany, who's going to talk about Coccolithophore and climate change. Before she starts, I will just give you a very, very small resume of Rosie, who did her PhD in Southampton in UK. With Sam Gibbs, Alex Poulton, and Paul Baum. And then she moved to for 1 year in Germany to Frankfurt, working with Jan Searle, if I pronounce it well. Then she moved to Nova Scotia in Canada, working for 1 year with Zoe Finkel, and went back to Frankfurt, having DFG fellowship or something like that, which is her current position. And I guess we are done on that. And so I thank her very much for doing the presentation today in NannoTalks. And I will just leave the microphone to her now.
Great, thank you. I'll just share my screen. Okay, does that look good? Yeah. All right, then. Thanks very much. So hello, everybody. Thank you for joining today. I've quite ambitiously entitled this talk Coccolithophores Under Climate Change, which, as I'm sure we can all agree, is quite an ambitious title for a 30-minute talk. There are literally thousands of researchers around the world focused on this very topic and studying various aspects of how climate impacts coccolithophores and other marine organisms, of course, because the impact of changing climate conditions on phytoplankton is far from a single cause and effect process, obviously. Firstly, we've got many different physical and chemical properties of the ocean changing simultaneously under climate change. And so we have many different drivers to contend with. And secondly, the impact of climate change on marine plankton actually involves many different types of impacts. So some may impact every marine organism, others maybe impact one group more than another, or just a few species are impacted. So many of you will be probably involved in researching the impact of specific environmental drivers on species physiology, for instance, through lab experiments. And these types of data are, of course, excellent for identifying cellular processes involved in physiological responses and the magnitude and variability of responses within important species. But a major challenge is scaling up those results to address the ecological impacts of climate change. So talking about changes to species biogeographies, changes in community composition or diversity, productivity, trophic interactions, these sorts of things. And ultimately, when climate change alters the balance of temperature, nutrient, chemical, and light conditions that phytoplankton need to thrive, their abundance and distribution also shift in response. And this leads to reorganization of community structures. And this has ramifications across food webs and for the biological carbon pump and the countercarbonate pump that are major driving forces of the global carbon cycle and key ecosystem functions. So to address these questions, there's been a lot of advances in recent years towards thinking about communities in terms of the traits that best characterize the role of different individuals in major ecosystem functions. And one of the most widely used traits, especially for plankton communities, is cell size. Now, most phytoplankton are single-celled organisms, which means that the size of their cell has a really strong influence on almost all aspects, really, of their physiology and ecology. And I've just shown a few examples here on the right of how some key rates and other traits scale with cell size or cell volume in phytoplankton. So, for example, all metabolic rates like growth and photosynthesis are related to size somehow, with smaller cells having higher metabolic rates relative to their mass. And cells of different sizes also have different nutrient, light, and CO2 requirements, for instance, amongst other things, because it affects the rate at which you can uptake resources from the environment and how much of that resource you need to maintain your essential cell functions. Now, because of the influence of size on physiology, size is also an influence on the ecological preferences of different species and their biogeography and their productivity. And this in turn means that the size structure of a community is a really important determinant of the role of different species and different phytoplankton functional groups in key ecosystem processes like the production and export of organic and inorganic carbon, as well as the complexity of food webs and how energy is transferred from primary producers like coccolithophores up the food chain. And typically, we find that communities in stable, low nutrient regions, such as subtropical gyres, for instance, are dominated by smaller cells, so the picoplankton and the nanoplankton, due to constant limited nutrient supplies. And this leads to complex food webs and highly efficient recycling of elements. Whereas in contrast, in more dynamic environments, we tend to see that the biomass of the larger phytoplankton plays a much bigger role. And this gives rise to shorter food chains and increasing material exported out of the photic zone. So understanding the basic mechanisms that govern the size structure of phytoplankton communities can give us insights into the potential impacts of climate change on the biogeochemical and ecological functioning of marine ecosystems. Now, for coccolithophores specifically, cells tend to range between maybe about 2 microns to as large as 30 microns, and at least across modern species. And this means that coccolithophores have a range of cellular biomass, so organic carbon. They're also characterized by a quite extraordinary diversity of morphologies. And this gives rise to a wide range of cellular calcite also across species. The main question is then if species distributions or their productivity change due to climate change, what are the biogeochemical and ecological consequences of those changes? Now, due to their size and morphological diversity, the answer to that question isn't necessarily that easy to figure out. So, for example, the most abundant species in a community isn't necessarily the biggest contributor to biomass or calcite, as was shown quite nicely by Chris Daniels and colleagues, who found that Coccolithophores pelagicus was the major calcite producer in the Arctic Ocean, despite only consisting of about 2% of the community. And that's because cells of Coccolithophores pelagicus are vastly larger in biomass and calcite compared to a cell of Emiliania huxley, which is numerically quite dominant in this area. And this prompts plenty of questions about the roles of different species in calcite production and export, spatially and through time. So if we're looking at a present day community, it's relatively easy to determine the size structure of the community and its biomass and its calcite. So if you're out taking a sample on a research ship, for instance, you can put your sample through a flow cytometer to get the overall size structure, or you can filter your sample and look at it under a microscope and just measure all the cells that you find in that community, such as this E. Hux bloom. Kindly, this picture was given to me by Kyle a few years ago. I thought it was a nice example. And we can determine organic and inorganic carbon, of course, analytically in the lab through several methods. I'm sure many people listening do this quite routinely. But today we're going to take a microscope-based approach. So just as an example, here's a subtropical gyre community from the South Atlantic that was sampled during 2004 on one of the Atlantic Meridional Transect cruises. And by measuring the size of all the cells in SEM images from this site, we can determine the size structure of that community, which consisted of 30 different species. And as you can see here from this size histogram, the mean cell size is around 7 to 8 microns and ranges between around 3 microns, the smaller cells, and up to about 20 microns in the larger cells. Most cells tend to be between about 4 and 9 microns. Using morphometric measurements, we can transform the size structure into how much biomass and calcite a community of that size structure and that composition actually represents. And to do this, we use two different techniques. So firstly, to transform from size into biomass, we can look to lab experiments that have shown that biomass scales proportionally with cell volume, which we can determine from measurements of cell size using microscopy. Now, mostly to date in our work, we've been using a volume to carbon ratio published by Mendendoyer and Lassard, which is shown here as this blue line on the graph. And that's based on a data set of many different species of phytoplankton, only a few of which were coccolithophores. Recently, though, a new relationship was published where the cell size and biomass of a range of different coccolithophore species that had a range of cell sizes was investigated. And the data from that experiment suggested that coccolithophores may actually be less carbon rich than suggested by the relationship of Mendendoyer and Lassard. And actually the change in the slope of these different relationships makes quite a difference in the biomass of cells of the same size, depending on which relationship you're going to use. So if you are doing experiments and measuring POC or biomass, then please do also measure cell size about the coccosphere so we can build on these data over time. In terms of calculating an estimate for how much calcite would be in a community like this based on morphometrics, we used an approach from Young and Zaveri, which I'm sure many of you will be very familiar with already. The principle works on the fact that the calcite of a cell is a function of how much calcite there is in a coccolith, and then how many coccoliths there are that make up the coccosphere. Now, coccolith calcite is a function of coccolith length cubed, coccolith volume, with a species-specific shape factor applied that adjusts this volume to a more accurate volume depending on the morphology of the species, and also times by the density of calcite. Now, of course, each stage of this equation already involves some level of simplification. So coccoliths on the same cell do show some level of size variability. And within the same species, there's obviously also plenty of size variability. As you can see here, just put together some data for Coccolithophores pelagicus from some lab experiments compared to some field data compared to what Coccolithophores pelagicus looks like in fossil assemblages. And you can see there's a broad range of different size distributions there. The shape factor applied also is a simplification for the range of shape factors across individual coccoliths in the same species. So worth bearing that in mind. And in terms of the number of coccoliths per cell, this is relatively easily counted through microscopy, but coccoliths per cell is also variable, particularly depending on the point at which the cell is in the cell division cycle. So recently divided cells will have fewer coccoliths than cells that are grown for a while and are ready to divide again. And this can lead to differences in overall population coccoliths per cell, depending on whether more or less cells are growing exponentially or not. So just some points there to bear in mind going forwards. But if we apply these two approaches to this modern community example that I've shown, then measuring coccolith size, cell size, and the number of coccoliths per cell on each cell that we come across, we can calculate their pick and POC. And in doing this, we can transform our size structure into a biomass distribution. And you can see that even just one or two very large cells like we have here, they can have a disproportionate impact on the total biomass of the community as their volumes are just so large. And in comparison, the higher abundances of smaller cells in the community are contributing way less overall to community biomass. We can also look at community calcite, which again is more skewed towards medium-sized classes. And in this community, community, species of these larger sizes have more calcite per cell than species that are dominating the smaller size classes. So we end up with this shift towards the middle. So that's a look at a modern example. But for me, and I'm sure for many of you, we are also very interested in what we can learn about the impacts of climate change on coccolithophores from the fossil record as well. Because Many plankton groups have really exceptional fossil records that capture the response of the whole community to climate change in all of its ecological and environmental complexity, and on a range of timescales, and including climate states that just do not currently exist or don't exist recently. So we don't have other analogs for them. The fossil record is a really rich resource for us to draw on for investigating how climate affects coccolithophores. Now, the challenge is that whilst we might have pretty much the same questions in mind of the fossil record as we do of modern communities under climate change, fossil data sets are usually not that comparable to the types of physiological and ecological data sets that we generate from living species. And that's simply just because we can't always measure the same things. So we end up with very similar questions, but with different tools to answer them. Now, morphological traits are a great exception to this, though, because identical measurements can be made on both living and fossil cells. However, generating size records through time from the fossil record of coccolithophores faces a big challenge that I'm sure is very obvious to all of you. But when you look at a fossil assemblage, you will see that basically all you have is individual coccoliths, thousands of them. And of course, we can do detailed morphological studies on the coccoliths themselves, but intact fossil coccospheres are comparatively very rare. And this is potentially a problem because without intact cells, it's really difficult to be sure what the species looked like when it was alive. And we lose a lot of really fundamental information in the process, including cell size. For some species, we can use modern descendants to guide us, such as perhaps fossils of Gophairocapsa or Coccolithophus or Chalcodiscus, things that we have in the ocean today. But most fossil species don't have particularly close modern relatives or morphologically similar representatives that we can work with. And this could mean that we end up over or underestimating cell size in these taxa. And it's certainly a challenge sometimes to be sure if a species, for example, is polymorphic or not in this instance, if we don't have any intact coccospheres to go for. But all is not lost. There are plenty of reports in the literature of coccospheres that have been found in a range of different time periods and different localities. I've just thrown up a few examples here, but you can find plenty others on NannoTalks if you're looking at particular species. And particularly work done over the last decade or so on sites of exceptional preservation has revealed quite literally thousands of fossil coccospheres from many genera that we can use to understand cell size in the fossil record. So with fossil coccospheres, such as this beautiful Eocene Coccolithophus example here, we can take measurements of cell and coccosphere size, coccolith size, and number of coccoliths per cell. And this gives us completely comparable data sets that we can collect from both modern and fossil coccolithophores. And this means that we can quantify the relationship between cell size, coccolith size, and number of coccoliths per cell for many species or genera. And this crucially allows us to model cell size from coccolith size, which is something that we can, coccolith size is something that we can measure in pretty much any sample. So you can see from our working database here of coccosphere geometry, we've analyzed more than 4,000 coccospheres and counting, and that overall the relationship that larger cells have larger coccoliths that was first presented, for example, by Urantia Hendricks in her 2007-2008 work. This generally holds for a much larger diversity of species that we've come across. However, you can see that there is a lot of spread in this relationship, and that's because some species have fewer larger coccoliths and other species have many small coccoliths, and there's plenty of variability in the degree of coccolith overlap on a coccosphere and many other factors. But for genera or particular morphogroups that we might be interested in, we can derive relationships that calculate cell size from coccolith size and number of coccoliths per cell. And to do this, firstly, here's an example using Coccolithophus in the late Eocene. we use the relationship we just derived for the species or whatever morphogroup you're interested in. We calculate the cell size that would result from any combination of coccolith length and number of coccoliths per cell. So here you can see for this coccolithus example that as the coccolith sizes get bigger and there's more coccoliths on the coccosphere, the cell size correspondingly gets larger. But of course, not every combination of coccolith size and number of coccoliths per cell is actually realistically going to occur in our sample. The size of the coccoliths or how many there are in a cell might be out of range for that species generally, or they might just not occur in that time interval or at that location. So we measure the size distribution of coccoliths of the species in our sample, and we apply the range of number of coccoliths per cell that we're seeing in fossil coccospheres and the range of coccolith sizes that we see in our sample to predict how likely each combination of coccolith size and number of coccoliths per cell is to exist in our sample, which you can see here. So you can see that actually we don't have, I guess, a real result for the very largest and the very smallest combinations of coccolith size and number of coccoliths per cell, which is to be expected. But the sizes that occur in the colored data points here do exist, and some of them are more likely to show up depending on the coccosphere geometry of this sample. And by matching up these results, we can create a cell size distribution for this species that's specific to this time interval and to this sample. But community structure, size structure doesn't depend solely on the size distribution of each species, but also on the relative abundance of that species in the community. So final steps are to normalize the frequency of each species reconstruction or morphogroup size distribution before making sure that the area under the curve is equal to the relative cellular abundance of the species or morphogroup in the sample. So this method's a little bit complicated. It's complicated to visualize. So if you are particularly interested in this approach, then please check out our paper Gibbs 2018 or get in touch for more details. I'd be happy to talk through it in more detail. So not sure how we're getting on. Okay, so to finish up for today then, I want to show you a few examples of this process in action in the fossil record, where the size structure of fossil coccolithophore communities has been reconstructed to look at how climate change has had an impact on coccolithophores during the Paleogene. The first example I'm going to show you looks at the impact of the Paleocene-Eocene thermal maximum on coccolithophores. The second example contrasts the size structure of early Eocene peak greenhouse communities with late Eocene communities. So that's after several million years of global cooling and decreasing CO2 levels. And the third example will be just a brief look into my current project that investigates the size structure of coccolithophores at a single high latitude site in the South Pacific during the Oligocene. So we're going to span some important time intervals of the Paleogene and capture different climate states and also some different spatial resolutions. So starting off at the PETM, firstly, we're going to look at what happens between the background of the PETM in the lower three panels and the PETM recovery towards the top in terms of cell size structure in the first column and community biomass in the second two columns. Now, these data are from Shatsky Rise, which is an open ocean, low latitude gyre site in the Pacific. And you can see that there's quite a broad range of cell sizes that doesn't seem to show much variation through time in the first three panels there. However, this quite flat size distribution conceals the fact that it's the larger cells in the community that are making the strongest contribution to community biomass, as you can see in the center panels, even if they are present in lower abundances than some of the smaller size classes. Now, this first biomass column in the middle, that assumes a constant abundance through time. But if we weigh instead those distributions to the biomass yield that might be expected in a subtropical open ocean gyre site through the PTM, we can see that because there probably wasn't a particularly high number of cells per milliliter at this site, given that it is an open ocean gyre, we end up getting quite a flattened biomass distribution, but still with the larger size classes contributing most to that. If we look then in detail about the contribution of different morphogroups, different genera to this size structure, we can see that Coccolithophores, shown here in orange, is a big contributor to biomass, whereas Tawias, which is in this green color, is abundant, but only contributes minimally to biomass, really. And then the rare abundance of nanolith taxa like Discoasters and Sphenolithus and Fasciculithus, which is shown in gray here. And you can see particularly obviously in the center biomass reconstructions, they are contributing substantially to the overall biomass of this community. Now, the story through the PTM is quite different at Bass River, shown here, which is a temperate shelf site in the North Atlantic. And here, most cells are small, centering around 5 microns, with only a few larger cells. Again, Tawias is shown in this green color and Umbilicosphaera in this yellow color. And during the PTM recovery, particularly, they're really dominating that size structure. And even these low abundances of larger cells that you can barely even see in the cell size histogram, they balance out the they are overwhelmed by the abundance, the low abundances of larger cells. As we can see in the biomass reconstructions, again, we've got a much more flattened biomass distribution than you would expect by looking at the cell size distribution, because these few larger cells are really contributing so much biomass to the community. And during the peak and recovery of the PTM, the biomass associated with larger cells declines, which is effectively halving the total amount of biomass per 100 cells in the middle. If biomass is then scaled to the expected abundance of cells in the community, which is shown on the right-hand side, we expect that the intensification of the hydrological cycle during the PTM would increase the available nutrients at Bass River, and that would lead probably to a huge increase in total biomass through the recovery. Now, if we move forward in time a little bit, comparing community size structure between the early Eocene and the late Eocene, so here showing an average of results across 6 sites that are globally distributed because they all had broadly similar size structures. We can see that the cell size distribution in the early Eocene is also centered at roughly around 5 microns, as during this time interval, the genus Tawias, which is very small, was particularly dominant. And in biomass terms, the relatively low abundance of larger cells is again very apparent, and it flattens out the packaging of biomass across smaller and larger size classes. Now, if we compare this to the late Eocene, we definitely see some changes. So the size structure has shifted towards larger size classes, and mean community size is nearly doubled. And you can see this long tail of low abundance, very large cells come through. And that change is due to the extinction of Tawias and the origination and rapid size diversification of Reticulifenestra, which ranges from less than 3 micron cells to over 30 micron size cells. And the impact of that change in community structure on biomass per 100 cells is quite dramatic, with most biomass partitioned into size classes that are larger than 25 microns compared to the early Eocene that's much more spread out. So between the early and the late Eocene, global cooling and decreasing carbon dioxide levels were driving changes in water column structure and nutrient availability. And it looks like this is supporting the expansion of species into larger size classes where previously they might not have been competitive, or perhaps their resource requirements were not being adequately supported by the environment. Now, lastly and briefly, I just want to show some very preliminary results from a new site I've been working on in my current project, which is investigating the size structure of coccolithophore communities through the Oligocene. at a high latitude site U1553. And that site was drilled early last year on IDP Expedition 378. And similarly to previous examples, the first column shows the reconstructed size structure from the latest Eocene in the lowest samples through to the late Oligocene in the upper samples. And most of the cells are between 5 and 15 microns, with varying abundances of cells in larger size classes through the record. And these initial reconstructions suggest that mean community size might be broadly larger in the earlier Oligocene and decreasing towards the later Oligocene. But hopefully high resolution sampling that I'm currently working on should give us a better insight into whether that's the case and what changes in community composition are primarily driving any of those size changes. Now, although size structure is again skewed towards these smaller size classes, the lower abundance of larger cells can again clearly be seen to impact the biomass structure of these communities. But unlike some of the previously shown reconstructions, the bulk of the biomass seems to be more centered in intermediate size classes, so between maybe 10 and 20 microns. And this seems to be because many of the abundant species during the Oligocene actually have broadly overlapping size distributions. And so they're all clustering within similar size classes. So to wrap things up, I really just wanted to leave you with some points to think on. So firstly, that we can use cell size as a functional trait, not just in modern, but also for fossil communities. And this allows us to relate physiological processes to environmental changes and their ecological consequences. And in many of these reconstructions in the Paleogene, it gets you thinking about what is the advantage of being a smaller or a larger cell, and what are the reasons for that. So how do we link how small or larger cell is to interactions with the environment? And this enables us to track environmental changes and their impact through size structure of a community. And ultimately, the physiological responses of different phytoplankton species to climate change do seem to be reflected in their ecology, certainly. And so when climate change causes community reorganization, which it so often does, we need to be thinking about how new community size structure impacts ecosystem functions, like the role of different groups or different species in biogeochemical cycles through how much biomass or calcite they're producing and how it's distributed across different size classes. And with that, I'd like to thank the many colleagues who've been involved in this work over the last 10 years or so, particularly in current working group, Jens, who I guess is here. I think I saw his name pop up, and the rest of our working group. And of course, my colleagues from my time in Southampton, particularly, so Sam and Paul and Alex, Sarah, and they really got the ball rolling on the potential for coccolithophores and finding me beautiful material to work on over the years. So big thanks to them. And my current project is still ongoing. And a lot of what I've shown generally in the talk is unpublished and part of multiple projects in progress. So please, if you are particularly interested in one aspect or another, do get in touch with me by email to discuss it or to get an update on the current status of those pieces of work. So thank you very much for listening. I can't hear you, but is that just me? Oops. I can't hear you. I don't know if anybody else can hear you.
I can't hear him either.
Okay, there was a big beep and then I didn't hear anything.
Wait for it.
Ah, there we go. Hello.
Can you hear me? No?
Yes. Yeah,
Oh.
I can hear you now. Yeah.
I don't know, like my microphone doesn't work anymore. Does anybody? It was a beautiful talk, really interesting talk. I can't wait to see all these papers coming out.
Yes.
Does anybody have a question? Paul, I guess you open your microphone and maybe you have a question. Maybe not.
No questions. Paul knows this work too much, too close.
Okay, so I do have a question.
Mm.
At some point, you talk about the contribution of disquesters.
Yeah.
I wonder how you evaluate the amount of biomass they contribute, because there is no
Yeah.
so you probably have a trick to do to go through that. So
yeah, so discoasters, sphenoliphus, all these types of things that can be very abundant in certain sites through the Paleogene are frustrating because, of course, like you said, we don't really have any equivalent species that we can look at to know what they would have looked like. And sadly, we never find anything that resembles an intact cell in the fossil record. So you can check out the paper I mentioned, this Gibbs 2018, for the full details. I think it's in the supplement actually for these taxa maybe. But basically, we looked at the morphology of the nanoliths from the main discoaster species at this time. So for example, multiradiatus. And each lith does have some degree of curvature to it. And using that degree of curvature and basically like how big the cell is, how big the lith is and its surface area, you can approximately model how they could fit together to form a sphere. And we basically use that approach to estimate how large a cell would be relative to the size of the coccoliths. But with these taxa, I mean, there is a lot more uncertainty for sure associated with those reconstructions than there are for other species that we do have examples of what they look like. Also for some taxa, I mean, a lot of things that aren't placcolith taxa, we sadly don't really see as intact cells, but we do find collapsed coccospheres. So obviously it was one cell and all the coccoliths are there. And that at least gives us an idea of how many coccoliths would have been around a single cell, even if we don't have it intact. And that with the coccolith size allows you to make a pretty good estimate of what the cell size would have been. But yeah, there's challenges certainly for some of the taxa. Yeah.
Okay,
good point.
Thanks. Anybody has a question? Otherwise, I keep talking. Oh, Anita.
So, Anita, hi.
Hi. It's nice to see you.
Yeah, nice to see you too.
Can you go back to a figure, please? About,
yeah, which one do you want?
Cell diameters and more coccoliths per cells. It's a really colorful one.
Yeah. Let's see what we've got. This one?
No.
Nope.
It was like a purple reddish color.
Oh, in the kind of modeling part, this bit.
Yes, and the second one.
Yeah.
Yeah, okay. Can you tell me more about this method, what you used here?
Yeah, so unfortunately, the method part always seems to be quite complicated for describing in talks. So yeah, the left-hand part is effectively the modeled cell size that you get for literally any combination of coccolith size and number of coccoliths per cell based on the real relationship between cell size, coccolith length, and numbers of coccoliths per cell that was in that scatter plot, this one here. So this is obviously all the data. So it's a whole bunch of different taxa, but of course you can plot the same data set for just Coccolithus or just Coccolithus during one specific time interval, which I haven't got mocked up anywhere. But through that, you can make an equation that links all three parameters together. And so you can calculate cell size if you've got coccolith length and number of coccoliths per cell as an input. So that's what this grid does, basically. It takes any combination and it kicks out a cell size for every single combination. But not all of those combinations exist. So, I mean, here in the very uppermost top left-hand corner, I mean, you've got a cell with a cell with one coccolith that's 1 micron in size. I mean, that's just not a realistic solution. And at the other extreme, you've got a cell that's got a coccolith of maybe 20 microns, and there's 23 of them or something. It's going to be an enormous cell. So, you know, some of those outputs are just not real. So we have to then say, okay, well, what is the real distribution of number of coccoliths per cell in this species at this time? That's what's shown in the upper histogram. That's how many coccoliths per cell there are in the late Eocene in Coccolithophores coccospheres that we found. And we can go into a specific sample that we're interested in and say, okay, let's measure 100 coccoliths of Coccolithophores. What's the distribution of coccolith length then? And by combining those two real data sets, that's where we get kind of the results that do exist, and not just which ones exist or not, but also which ones are most likely, because more of the cells have those combinations. So that's the yellow colors that are shown in the middle. The ones that are blue, they can exist. There are cells that have those combinations of features, but there's maybe only 1 or 2, so they're less likely. And then we kind of basically match up the results. So how likely is it, and what size is that cell? And that's when we bring all of that data together, that's when we get this curve of cell size.
Right.
That makes
so
more
for the
sense.
whole Paleogene, then the middle figure is for the whole Paleogene.
The middle figure is just for the late Eocene in this case, just for this genus. And for a specific samples, you can just say, okay, well, for this specific sample, I will do the coccolith length just in this sample, which is what I have done in the Oligocene reconstructions that I showed you right at the end. That the coccolith size data for every single one of those reconstructions is measured on every single sample individually. So it's completely unique result to that sample. Or you can pool all of your data if you just want a more broad overview and say, okay, I'll measure coccolith length across a bunch of different sites, roughly in this time interval, to get a broader picture. So you can kind of take the approach that you want, depending on how detailed or specific you want your result to look like. Yeah. So it's quite really versatile. Yeah. Yeah, if you're interested in playing with it, the full details of the method are in this paper, the Gibbs 2018 one. We put it in as much detail as we could, so that people could have a look at it. Yeah.
Cool. Thank you very much.
Thank you, Anita. There were actually a question that I will read, but my English is that perfect that if you don't understand it, tell me. It said, thank you, Rosie, great talk. Coccolithos pelagicus has been considered as an indicator of warm oligotrophic condition in the Eocene, but today this species has a different ecological affinity. Based on your observations on Pelagicus, what could have happened to it, or can you see any change in the cell size?
Yeah, it's a great question. Coccolithos actually is one of my favorite species, and that's largely because it's just been around for the last 66 million years. So it seems to be quite versatile in how it can deal with environmental change, generally speaking. And you're quite right that, yeah, during the Eocene, for instance, Coccolithos is generally considered to be a warm water taxa, whereas now, if you look in present day oceans, you'll find that Coccolithos pelagicus tends to not be found in waters that are warmer than about 14 degrees. So it's more subpolar taxa in the North Atlantic, particularly. And we have another species or Morphe species, Coccolithos brudii, which is found in more temperate to subtropical waters, so warmer than 14 degrees. And they do look different morphologically, pelagicus and brudii. So I did actually ask Jeremy Young this exact question once, like, how did it go from being a warm taxa to a cold taxa? I think we had a great discussion, but I'm not sure we ever came up with a real answer, other than to say, I expect that just over time, it's quite a big species, it's quite heavily calcified, Coccolithos, and it might just be that with global cooling, for instance, over much of the Neogene particularly, we've had quite a change in in how much nutrients are available, water column structure, and generally speaking, those changes have just been more favorable for other species like the Nola habdaceae. They've become very abundant. They're smaller, which means they're much more competitive for nutrients. They can really exploit any changes in nutrient availability. Whereas species like Coccolithophores, they're bigger, they have high nutrient requirements. They maybe can't keep up with the same growth rates as something like EHUX over time. It could just be, I mean, this is really just me guessing at the moment, but it could just be that compared to warmer waters, they just are not that competitive compared to what else is there, but they found a niche in these subpolar waters. And in the meantime, a larger Morphe species of this same genus, we class it as a different species, but it kind of looks the same. It's just bigger and has bigger coccoliths. And that one's just found its niche in kind of more subtropical waters. So I don't really have a great answer. And I'm sure there's plenty of people here who probably can contradict everything I've said. But yeah, it's an interesting shift.
Thank you very much. Anyone else? Otherwise, I have a question, but I have plenty of questions. But if anybody else wants to have to ask any question? No? Okay, so I go.
Yeah.
So just for the audience, I've been in touch with Rosie lately based on the amount of data that she's done over the years. And so if I remember well, it was some of the data you sent me. I don't remember exactly which species it was, but I was surprised that there were like, I don't remember exactly that, like 5 or 10% of the coccoliths that were bigger than the cell diameter. Do you have any clue on that point?
Yeah, so this happens sometimes. Generally speaking, when you think about coccolith size in relation to cell size, the obvious thing to think about is, okay, well, the cell is a certain size and the coccolith is produced inside the cell, so it cannot be bigger than the cell. So it can be quite confusing if you find a cell where, okay, maybe there's some variability in coccolith size in the coccosphere, but you've maybe got 1 or 2 actually quite large coccoliths that are certainly bigger than the size of the cell. And first glance, you think, well, that's just not possible. But as I mentioned, back when I was talking about the calcite reconstruction part, should I just go back to, yeah, so here you can see, this is a range from a single strain. Actually, these might be field. No, I think this is a strain of Coccolithophores pelagicus. And so from a single strain in the lab, whenever you look at your cultures, you will find there's a range of coccolith size and a range in cell size. And this is because when a cell divides, its size effectively kind of halves, and it will have half the number of coccoliths. So by the time it's grown up again, that cell is much larger. It has a much larger volume that you could fill with a coccolith. And so a coccolith that's produced when the cell is at its biggest state and then divides, that coccolith is going to go on to one of the daughter cells. And so you might end up with a daughter cell where it's got one of these much bigger coccoliths than the cell actually is now. So that's the easiest explanation for why you can have a much larger coccolith than actually the cell size at the time. It's just to do with changes in cell size through the cell division cycle. It's not magic.
Okay, thank you. Actually, I've come to the same conclusion. To be honest.
I know you have, because I've seen your.
I wanted to have your idea on that point.
Yeah, no, I absolutely agree with the conclusions that you'd come to. Yeah.
So that's
reassuring.
That's good. Anybody else has a question?
Hey,
Anita has another question.
Yeah, I got one more, but it's just for fun. That have you been on the IADP cruise yourself?
Yes. So I sailed on this cruise last January. I think it was the last cruise that really went out before the COVID restrictions. We got back just in time. Yeah. So these are the samples that I looked at on the ship in these Oligocene ones. Yeah. It's been a fun project so far.
Yeah, it sounds pretty cool.
I encourage anybody to sail if they have the chance. It's really great.
It was in January in the Boreal region.
It was in the southernmost part of the South Pacific, so just south of New Zealand.
The other side. The other side.
Yeah, the other side. Yeah.
Okay.
On the edge of the Southern Ocean. Yeah. Yeah. It was basically a redrill of the classic DSDP 277 site that Shackleton worked on. That the first indicators of the PTM were identified in those classic papers. So it's a really cool site. Yeah.
Great. Does anybody have any comment, question? No?
No.
No. Wait. Oh. Okay. Sorry. No, it's I have 3 screens actually. So sometimes there is some stuff that popping around. So I guess there is no any more questions. So I would like to thank Rosie for a brilliant talk today. I remind everybody that actually it was recorded, and you may want to contact Rosie if you have questions. You may want to watch it. Well, you may watch it again before if you have some point. But your email address was on
I'm just going back.
slide.
Oops. There you go. If you want to contact me, the email address is there on your screen. Please get in touch. I'd love to hear from you.
I did it in the past.
Yeah, and I'm very helpful.
Helped me a lot on one project. So you go for it. She's really nice working with her.
And I obviously love talking about coccolithophores. So really get in touch.
And for the next talk, that will be in January. The date is not completely settled yet, but it will be probably it will be the week of the 17th, so the 3rd week of the year. And the talk will be done by, obviously I will lose the name, Fanny Montero. She will be talking about modeling and coccolithophores. And that's it. That's all we know for now. We will obviously send you any information through email like usually. And we thank again Rosie for a brilliant presentation today. And I thank all people that came today. And I hope to see all of you in a month. And I guess that's it. Oh yes, there is a Coco catch-up by TMS a bit later today. If you want to participate to that, you should ask Odysseus. Is the one planning it. I don't have any other information, to be completely honest. Thank you, everybody. Thank you very, very much, Rosie.
Thank you very much.
Wait to see you in the future, or work with you maybe one day, I hope.
Yep,
and thank you, everybody. See you.
Bye. Thank you very much, everybody.
Bye-bye.
Bye. Thanks.
Bye.